Good morning, students. The next topic that we are going to see in this research topic is sampling types. Okay, so we are going to see the different types of sampling. Usually this is a common question in your university paper, and also it is very important for you because next year when you do your research hospital oriented project, you need to remember this because you will be collecting samples, and you need to understand what type of sampling that you will be using. There are two major types, okay, two headings. One is probability sampling, the other is non-probability sampling. We already know sampling means selecting some people from the total population which you are going to include in the research. For example, if you are going to do research on cancer patients, then you cannot do research on all the cancer patients in the whole world. You have to take few people and do research and ensure that your research is applicable to all the people. So then your research will be valid. That's what we studied in the last class. So your sample type should be correct so that it represents the whole population. Even though you do research on a small number of people, it should represent, or it should be applicable to the whole population of people. Okay, so probability sampling uses random sampling methods, okay, and non-probability uses non-random process like the researcher judgment or convenience sampling. So random means there is no involvement of the researcher. It is random process. It is an automatic process, and nobody knows who will be selected in the research. So that is the advantage of random. So for example, if there are 100 people in Karigiri college first year or second year, then if we are going to do research, we can select any 10 people. So each person in the 100 people will have equal chance of becoming part of the research. So every single person has a chance. For example, if you are picking lots, like rolling the dice, each time you roll the dice, there are 6 sides in the dice. So any number can come, 1 can come, 2 can come, 3, 4, 5, 6, any number can come in the dice. Do you know which number will come? No, it is random. So that is the meaning of random. So probability sampling is like that. Any number can come, any person can be selected for the research. So there are different types in probability sampling itself. The first one is simple random sampling. Okay, simple random sampling means this is just like the rolling the dice. Or another method is you can write 100 people's name and put it in a box and shake the box and pick out any 10. So you don't know which 10 people you will select. So every single person has an equal opportunity to be part of the research. So that is simple random sampling. So it's like just writing each person's name. If you have 100 people in the college, all 100 people's names is written, put it in a box and just pick out 10 names. So those people will be part of the research. This can be done through computer also. Like if it's 100 people, you can write their names. If it's 10,000 people who are, if you are having a study population of 50 lakhs, of which you are selecting 10,000 people for the as samples, then you cannot write 10,000 names. So instead, if the names are entered in the computer itself will randomly select 10,000 people out of so many lakhs. So that is simple random sampling. Also there is a calculator called T182 which can be used to generate the random numbers. So this is simple random sampling. Hope you understood. So it's like the rolling the dice. Nobody knows which number will come. So like that, of the study population, nobody will know who is going to be selected as samples. Every person has an equal chance of being part of the research. So that is simple random. Then stratified random sampling. Stratified means there are different groups or subgroups For example, we take 100 people for a study on coronavirus. That's an example. Okay, so coronavirus can affect anybody. It can be a male, it can be a female, it can be a child, it can be an adult, it can be a old person. So we stratify so that every group is represented. We group the people who study population to males, females, males less than 5 years, males between 5 and 50 years, males over 50 years, females less than 5 years, females between 5 to 50 years, females over 50 years. And then we select from each strata, we select samples in a random manner. Previously we studied about random manner. The same random manner, after we stratify the people into different groups, we select so that every group is represented in this study. So that is stratified random sampling. Then systematic sampling. Systematic sampling means every nth number. This is common in your school. If you had 100 students, they will make you all stand in line and say 1 2 3 4, 1 2 3 4, 1 2 3 4, and then all the ones gather together, all the twos gather together, like that. So like that we can pick out every nth participant from the list to be part of the study. So if there are 10,000 people, if you want to select 100, then you will, every 100th person you will select for the study, then you will get 100 people. So that is systematic sampling. It's a systematic manner in which you are selecting samples for the study. Next is cluster random sampling. Okay, it is very similar to the stratified random sampling, but only difference here is you will actually take people from different clusters. Usually it is mainly if you are studying villages. For example, if there are 100 villages in Katpadi and 100 villages, you want 5 people from each village, then it is called cluster, because each area you are picking out people, or even each village or even each city, based on geographic location you are picking up people. That is cluster random sampling. Then multi-stage random sampling is a combination of all the above 4 methods. It is systematic, it uses cluster random method, it uses the stratified method, and the simple random. All four together form the multi-stage random sampling. There are advantages of each of these and some disadvantages also. So let's see the advantages and disadvantages. The cluster sampling advantages: it is convenience and easy to use. The advantage of simple random sampling is it represents the whole population, because you are randomly selecting. There is each person has equal chance of being selected, so there is no bias when you select the samples. The next is stratified random sampling, because you create layers or stratify. Stratify means you mark different groups and then select from different groups. So every group has a equal chance of getting selected few numbers. So I gave the example of coronavirus. So youngsters, males, middle-aged males, old males, young females, middle-aged females, old females. So every group has equal number of people representing in the study population. So that is stratified random sampling. Finally, the advantage of systematic sampling is because every nth person is selected. So just like we counted 1 2 3 4, so there is equal chance of being selected, and this is advantageous. Okay, and here also it represents the whole population because there is random selection. The disadvantage is in cluster sampling, if it is not a homogeneous group, then it does not work. Not homogeneous means not similar to each other. When you select people, they should be similar to each other for the study. So if they are not homogeneous, then cluster sampling will not work. It would be better to use random sampling or other methods. The simple random sampling method is time consuming. If, as I already told you, if you need a large study population, then it is not possible to do the sampling manually. You have to use the computer. Even in the computer, you have to enter the data of all the people, and then the computer will pick out randomly. So it is actually a time consuming process. Then again, stratified random sampling is also a time consuming process, because again you have to stratify into different groups, so it takes a long time. And systematic sampling again is not very random, just like the simple random sampling. So one major disadvantage of all this probability sampling is it takes time and it is costly. But the advantage is it is one of the best methods of sampling compared to non-probability sampling. Okay, so what is non-probability sampling? So non-probability means there is no probability. It is more like a selection by your mind. So it is not a probability. Okay, so here the odds of any member being selected for a sample cannot be calculated. Okay, it is the opposite of probability sampling, where you can calculate the odds. In probability sampling, suppose there are 100 people, then if you want to take 10 people for the study, then you can calculate the odds as 1 by 10. So there is a 10% chance of any person being selected for the study. Out of 100 you are selecting 10, so 10 by 100, that is 1/10. 1/10 chance or 10% chance is there for a person to get selected. In non-probability sampling, there is no way you can calculate the odds of the person. You cannot calculate the chance by which a person can get selected. So the advantages and disadvantages are: advantage of the non-probability sampling is it is time, it takes less time and it is cost effective. That is, you can easily select the candidates for the non-probability sampling. So it saves time for you and it is also cost effective. Most of you will be doing this type of sampling because it's easier for you, especially when you do your study for your college purposes. Non-probability sampling is easy, because otherwise you will spend a lot of time just in selecting the samples. So here the disadvantage is you don't know whether the people you selected for the study, they are representative of the whole population. Okay, so for example, if I need to select 2 people in your batch, so if I close my eyes and select 2 of you, then it could be that both the person are males or both the person are females. That will not be representing the whole group. Whereas if I open my eyes and select 2 people, I can select 1 male and 1 female. So that represents every part of your class. So that is the disadvantage of non-probability sampling. You don't know who you will select and whether it represents the whole population. So there is a chance that you cannot calculate the errors, which we will study later. So this is the disadvantage. But as I said, the advantage is it is less time consuming and less costly. So it saves you a lot of time and cost. So we'll see the types of non-probability sampling. What are the types of non-probability sampling? Convenience sampling. This is one of the simplest methods, simplest sampling. So what you do is you just select whoever is convenient for you. So for example, if you want to select 10 people with coronavirus, you just go to the hospital and start selecting patient number 1, patient number 2, patient number 3. You first 10 people who come to you, you select those people and do your research. So it's very simple. You don't need to calculate anything. You don't need to sit and take every fifth person or every 10th person. There's nothing like that. You don't need to type their names in the computer and then you write a program to select the people. You just go pick out any 10 people you find is convenient. So the disadvantage, as we already saw, you don't know whether these 10 people will be males or will there be 50% males, 50% females. So in the previous probability sampling, we stratified male less than 5 years, middle-aged male, old male. So here you don't know which person will come. It could be that all 10 are females, all 10 are more than 60. Then it will not represent the whole study population. So that is the risk of convenience sampling. Then the next one is haphazard sampling. Convenience sampling, you just picked the first 10 people. In haphazard sampling, you just close your eyes and just select 1, 5, 8, 11, 15. So there is no method in this. You just randomly select some 10 people. So that is why it's called haphazard sampling. Then the next, so because it's a haphazard thing, there is no way you can calculate whether a person has the odds of being selected. That's why in non-probability sampling, you cannot calculate the odds of a person being selected for research. The next one is purposive sampling. So purpose, as the name indicates, there is a purpose for selecting a particular person. So you decide who you are selecting based on the purpose of the study. So for example, if you want males who are less than 50, then you wantonly select males who are less than 50. So that you know why you are selecting and how you select it. So it is purposive. So there is no selection by chance. It's selection by purpose. It's the opposite of chance. So the disadvantage of purposive sampling is there is a bias. So you know as a researcher who you are going to select. So the risk is that you will select people who you think will benefit you will benefit as part of the research. So that is why purposive sampling is not very beneficial in research. Then expert sampling. Here in expert sampling, there are experts in the particular field. So for example, if you are doing research on coronavirus, you select an expert or a group of experts who then they decide who are going to be part of your research. For example, in your third year, if you are going to do a field research on leprosy, then you find 2 or 3 experts in leprosy in our hospital and you tell them to select the samples. So instead of the researcher selecting the samples, the experts of the field select the samples. So that is expert sampling. Then the next type of sampling is heterogenic or diversity sampling. So here you should know the meaning of heterogeneous and homogeneous. Homogeneous means same kind of group. So for example, all females, all males. That is homogeneous. Heterogeneous means a mix of male and females or a mix of young and old people. So that is heterogeneous. So in heterogeneous sampling, without a probability, in the previous probability sampling, stratified sampling was there. It is this heterogeneity sampling is similar to stratified sampling, but you select on your own. It is not a random selection. Okay, so the researcher selects people for the study based on their heterogeneity or diversity. So instead of it being a random selection, a purposive selection by the researcher. So that is heterogeneity sampling. Then model instance selecting. So here the typical members are selected from a set. For example, what we mean by typical members is, if you are doing research on leprosy, then you select people who have all the deformities of leprosy. For example, eye, hands, feet. Then they are typical of a leprosy patient. Whereas there are leprosy patients who do not have any deformity, they are normal. So you do not select those people, because then they will not look like leprosy people. Okay, they will look like normal people. So you make sure that you select samples that are typical of a particular disease, or they have the maximum effect of a disease. Those people you select for your study. So here again, this researcher can have a lot of bias when they select the sample. That is why non-probability sampling has less value in any research. The next one is quota sampling. Suppose you want 50 people in your study, of which 25 are men and 25 are women. Then you make a quota. You select one after the other, men one after the other, women, so that you have 25 in each group. Whereas if you do a random sampling like previous probability sampling, then you won't know whether males are selected, females are selected. So here you do a quota sampling, so this has less value. Finally, snowball sampling. This is 3 mark or 5 mark in your exam. So here the research participants themselves recruit other members for the study. Suppose you want 100 samples for your study on drug addicts. Can you find out 100 drug addicts in Vellore? No. It will be very difficult, because you don't know anybody who is taking drugs. So what do? You go to one drug addict who you know, then tell that person, see, you know a lot of drug addicts in Vellore, you select the samples for me. So the sample that you selected, they go out and select more samples. So that is snowball sampling. So here research participants recruit other members for the study. So here usually why we do snowball sampling is when we find it difficult to select samples on our own, we use this method. So if you want leprosy patients, it's easy for you. You go to the OPD and you select 100 people who are affected by leprosy. Either you take every 5th person who are affected by leprosy, or you go to the records and take anybody. It's easy to do that type of study. Whereas if you're doing study on prostitutes, or suicide attempted people, or drug addicts, it's difficult for you to find samples. So you go to one sample that you know and tell that person to find 100 more people. So that is snowball sampling. Hope you understood these different types of sampling. So here is a pictorial representation. If they ask sampling or types of sampling for a 10-mark question, then you can draw this diagram. So sampling has 2 types. One is probability sampling, other is non-probability sampling. In probability there are 4 types: simple, systematic, stratified, cluster. That is simple random sampling, systematic random sampling, stratified random sampling, cluster random sampling, and all these together form the multi-stage sampling. So here all these methods, in probability sampling everything is random. There is nothing by chance, it just happens randomly. So that is the advantage. Only thing is, this probability sampling is time consuming and very costly. But the advantage of probability sampling is it has more value in the research field. The non-probability sampling is, there is quota sampling. So you fill the quota. As a researcher, you decide who fills the quota. Then purposive. Purposive means heterogeneous purposive. That is, if it's different types of groups, then you decide who you select for the this thing. So it can also be typical purposive or extreme case purposive. So different kinds of purposive sampling. Then volunteer is snowball selection. Self-selection is the person themselves volunteer to become a part of the study. And then haphazard, I told you there is no method, you just select whoever you feel like, that is haphazard. This is a convenience sampling. The disadvantage of non-probability is there can be more bias, because the researcher plays a major role in selection of the samples for the study. So you will select people who you think will be positive for your study. So that is the disadvantage of non-probability sampling. The advantage is it is less costly and it takes less time. So that is the advantage of non-probability sampling. Okay, hope you understood the sampling, types of sampling. Next class we'll see the sampling techniques, sample size and its determination. Thank you.